Hi, and welcome to my presentation on the microbiology of the built environment, which would be things that we built. So we'll start with bioremediation. In bioremediation, you're using microbes to degrade pollutants that we have released into the environment. So this isn't necessarily like a built environment where we kind of created it. Usually it would actually be, more like the natural environment, but we have polluted it. So it bears, it bears the marks of our presence. Bioremediation has the potential to really help us effectively and hopefully not too expensively degrade a lot of different pollutants. It's a really great way to clean up anything that has spilled or been released into the environment, as long as there's a microbe that is able to metabolize that pollutant and degrade it. But in actual practice, we don't use bioremediation as much as we might, mostly because we don't know enough yet. So for the most part, we have used bioremediation pretty extensively to clean up oil spills or leaks. And we've also begun using it to degrade chlorinated compounds and uranium. Uranium would be mostly released during mining, and chlorinated compounds are found in a lot of different products actually, but prominently in pesticides. When you use pesticides, it's almost impossible to prevent them from escaping into the broader environment. As we learn more, we will be able to use bioremediation more to clean up more types of pollutants in more situations. Actually doing that means that you need to have a strong understanding of the microbiology of the microbe that you're trying to use to clean up this pollutant. You need to know also a lot about the microbiology of the environment where the pollutant has been released. You need to know what types of microbial communities live there, what they're surviving off of nutrient-wise, how the pollutant will be affecting them. And when you introduce this new microbe, you need to understand how that microbe will interact with that local community. Alternatively, a lot of the time you actually would not introduce a new microbe to degrade the pollutant. You would just figure that there are some representatives, or there is a population for that microbe already in this local microbial community. And then you would be probably injecting extra nutrients for it to favor its growth. And if you're going to do that, you also, of course, need to understand how those nutrients will affect the other microbes in the community. If you don't have a strong understanding of all those potential interactions between the pollutant and your chosen microbe and any nutrients you inject and the local microbial community, what can happen is that the bioremediation might fail. You might not degrade the pollutant or not as much of it as what you wanted. Or you might actually end up producing additional compounds that you didn't intend to that could be toxic as well. In bioremediation, the goal is sometimes to degrade the pollutant, meaning breaking it down, usually into some type of a form that is less toxic, or hopefully not toxic. But if that can't be done, then the goal is to just kind of contain the pollutant there where it is, basically prevent it from entering the groundwater and spreading to contaminate more areas of the environment. So for some pollutants, you can actually degrade them to a non-toxic form, or at least a less toxic form. For others, there's not necessarily a way to reduce the toxicity of it, but you can at least keep it there so that it doesn't poison the groundwater. A lot of pollutants are what we call xenobiotics. A xenobiotic is any compound that has been made by humans that does not exist in the natural world. So things that we basically created and invented. It's kind of hard to degrade xenobiotics using bioremediation in general because microbes would not normally have lived in environments that have these compounds in them, since they didn't exist before we made them, which probably means they haven't even existed for that long on the planet. So it's just hard to find microbes that would have the type of metabolism that allows them to degrade those xenobiotics. So, you know, it is possible for some of them, but just not for all of them. So with organic pollutants, well, organic or inorganic pollutants actually, just natural pollutants, it's very common to have microbes that can degrade them, including, you know, to a non-toxic form. For a xenobiotic, you might not be able to degrade it so well. Maybe it will still be in a toxic form. Maybe you're just trying to trap it in that environment so it doesn't spread. Maybe you can't really do anything with it at all if there is no microbe that can interact with that compound and catalyze reactions involving that compound. Actually, eventually, there should be microbes that are able to degrade, you know, any of these xenobiotics. It's just that, you know, it takes time for those functions to arise by evolution. So we'll start with the bioremediation of uranium. You would find uranium polluting the environment in places where uranium was mined, of course, but you can also find it in places where uranium was processed or stored. And actually the pollution is usually worse where it's been processed or stored, just because after the uranium has been processed, you're kind of concentrating it during that process. So you're going to have more of the radioactive uranium in processed uranium ore compared to raw uranium ore that's just come out of the ground. When you're bioremediating uranium, the basic goal is to convert the U6+ ion to the U4+ ion. Whenever you have uranium in the 4+ ion form, it will generally be incorporated into a mineral called uraninite, which is not a water-soluble mineral, so it's pretty stable. The U6+ ion is water-soluble, so if you have uranium in that form, it can enter the water, it will be washed away from the site, enter the groundwater. It can end up polluting, you know, of course, like rivers and streams and the ocean, but your drinking water as well. So that's not positive. So our goal here is just to trap it on the site by incorporating it into uraninite as the uranium 4+ ion. With both of those ions, they could be radioactive. So whether or not uranium is radioactive depends on which isotope of uranium you have. It doesn't depend on what ion you have. So they can both be radioactive and dangerous. It's just that one will easily leave into the water and the other will not. Once you have converted uranium to the U4+ ion, you still need to keep monitoring that site because there are certain conditions in which it can oxidize back to the 6+ ion, especially if you have oxygen around. Also, there's some other chemicals that can oxidize it spontaneously as well. So you would need to monitor the site, make sure that those, like oxygen and those other chemicals, are not present, so that you know that the uranium is remaining stable. If they are present, honestly, I don't know what happens then. Maybe you try to do the bioremediation over again. There's three main genera of microbes that are used for this process. You have Shewanella, Geobacter, and Desulfovibrio. All of those bacteria would be using either hydrogen, molecular hydrogen, or some type of organic compound as an electron donor, and then they're going to use the U6+ ion as their electron acceptor and oxidize it or reduce it to the U4+ form. So usually when you're doing this, you're going to figure that there are some microbes from those genera in the area already because they frequently live in the soil. So all you need to do is provide them with an electron donor to kind of stimulate their metabolisms. If they have more electron donor around that they're using, then they're going to need also to use more electron acceptor, so they'll end up using more uranium and converting it to the U4+ form. So that process would actually be happening even without your intervention. It's just that when you inject extra nutrients for these microbes, you're kind of giving them a growth spurt in a way. Their metabolism will speed up and the process will just go a lot faster. In the picture here, this is a sample of the uraninite mineral that you're trying to convert everything to. And here's a site that had uranium pollution in it, and it's being cleaned up in this way. So what they're actually doing is injecting organic electron donors into the ground to be used by these microbes for their metabolism so that they can take up more U6+ as an electron acceptor and convert it to U4+. So from these tanks, they actually have the electron donors stored in those tanks or coolers, and they're piping it into the ground. Next, we'll cover the bioremediation of oil. Oil consists of hydrocarbons, which is just long chains of carbon molecules linked together and all studded around with hydrogen. So nothing in it except for just carbon and hydrogen. Some of them are a chain, some of them will actually form rings, so some of them can have like a more complicated structure. They can also have branches if they're a chain, but they're always just carbon and hydrogen. So, of course, we have a lot of extraction of oil from the environment, from deposits deep underground, sometimes beneath the ocean. So you have, of course, the places where it's actually being extracted from the ground, and then the oil will be pumped somewhere else, either in a pipe or, on a ship, and then it's going to be processed and stuff as well. So anywhere along that process, you have the opportunity for a leak or a spill, which means that it's really not uncommon at all to have oil being released into the environment as a pollutant. And of course, sometimes we have a really spectacular catastrophe where a ridiculous amount is released into the environment. It can always be degraded by bioremediation, which isn't going to be like a complete fix or an instant fix, but it does clean up the environment much faster than without the bioremediation. When you're bioremediating oil or hydrocarbons, the end goal is to break them down into carbon dioxide. So you're going to completely oxidize the hydrocarbons to CO2. There's any number of microbes that can do this. Hydrocarbons have been around for a very long time, of course, so lots of microbes have evolved to be able to use hydrocarbons as electron donors for their metabolism, which produces either, shorter, smaller hydrocarbons or, eventually carbon dioxide. So you have aerobic and anaerobic microbes that can do it. A lot of them are bacteria, but you also have fungi and even some green algae that can do this process. When we're using them for bioremediation, we prefer to use aerobic microbes because their metabolism is faster. So they'll degrade the hydrocarbons to carbon dioxide faster than anaerobes will. Usually anaerobes would be coming into play in kind of I guess a bad way. Sometimes you have some of these hydrocarbon metabolizing anaerobes get into places where oil is being stored or pumped, and they will end up degrading the metal walls of whatever tank or pipe they're in, which leads to leaks, of course. In general, when you're bioremediating the hydrocarbons, you're not going to add bacteria or any microbe to that area because there are so very many microbes that metabolize hydrocarbons. You just kind of figure that there's some around. What you're actually going to be doing is just adding extra nutrients to help them work faster. So if you add nitrogen and phosphorus, then these bacteria, well, mostly bacteria, will metabolize the hydrocarbons much faster than they would otherwise. Again, they're getting a boost to their growth. Also, if it's warm, that helps also. So you could kind of try to warm the area up if it's a real cold place. If you have added nitrogen and phosphorus to the area of the spill, about 80% of the spill can be degraded within the first year to carbon dioxide, so essentially removed from that environment, which is really good. Sometimes, though, you might have some of that oil leaking down into the sediments during that year, which would kind of, it delays the bioremediation and it just prevents it from being quite as effective overall as it would have been otherwise. You're just kind of having that oil escape into the environment. So even though there's any number of microbes that can degrade hydrocarbons, there are certain microbes that degrade like certain hydrocarbons that are like extra hard to degrade. So the hydrocarbons that have like an extra complicated structure, like a lot of branches or a lot of rings in their structure, those are kind of hard to degrade. There's actually not that many microbes that can break them down, and it's only certain ones that can do it. In particular, the genus Sphingomonas has species that are able to degrade hydrocarbons that have multiple rings in them, which we call polycyclic hydrocarbons. So that's why that's a really useful genus for bioremediation. In the picture, you can see a section of a bank where oil had been spilled. This part where it's like brighter is where they had injected nitrogen and phosphorus to speed up bioremediation. And these areas where it's darker did not get any extra nutrients, and so it's still dark with oil there, basically. So you can kind of see visually like how much faster it is to get rid of that oil if you inject extra nutrients for the microbes that can degrade it. Finally, we'll talk about the bioremediation of xenobiotics, which are those compounds that we basically created from scratch. Or, not necessarily from scratch. We probably might have modified an existing compound, but just a compound that doesn't exist anywhere else other than where we made it. So there's a lot of xenobiotics, and a lot of them that end up as pollutants, but some of the most important sources of xenobiotic pollutants are pesticides and plastics. Plastics are especially tricky because they are polymers, which means they're really long chains of lots of repeating subunits. And in general, polymers are always more difficult to degrade than things that are not polymers or that are shorter. And then when you add in the fact that most of these plastics are made out of xenobiotics, so things that don't exist in nature, it gets really hard to degrade plastics. For any xenobiotic, the more closely it looks like some compound that does exist in nature, the more likely that it's going to be able to be biodegraded, or the more quickly that process will go. So if you show a microbe a xenobiotic that kind of looks like something that it already metabolizes, it's much more likely that the microbe will be able to do something with it. If the xenobiotic looks nothing like that microbe has ever seen before, probably it won't be able to do anything with it. One genus that's really important for bioremediating xenobiotics is Burkholderia, which is an aerobic genus. So all of the members of that genus would be requiring oxygen for their metabolism, using it as a final electron acceptor. Burkholderia is able to dechlorinate a lot of different compounds. So a lot of these xenobiotics are going to have extra chlorine atoms added to them, and those chlorine atoms are making them kind of extra toxic. So Burkholderia will take off the chlorines, which is already reducing the toxicity of the compound, usually by a lot. But actually, in a lot of cases, it can go further, and after those chlorine atoms are removed, it's able to kind of chop the compound up and use the pieces of it as electron donors, producing like even simpler and even less toxic waste products. So that's for aerobic dechlorination. You also have anaerobic dechlorination that's performed by a number of different microbes, which would be, of course, an anaerobic process, so it does not require oxygen. In this case, the xenobiotic would actually be used as the electron acceptor for anaerobic respiration. And in that process, the chlorine atom would be kind of removed from the compound. So you have microbes that can do that for xenobiotics that are chlorinated, but you also have a lot of xenobiotics that are brominated or fluorinated, so they've had a bromine or a fluorine atom, one or more, added to them. So you have as well microbes that can do anaerobic debromination and anaerobic defluorination, where the compound is used as an electron acceptor, and that process of accepting the electrons onto it and reducing it kicks off the bromine or the fluorine or the chlorine. So this process is not really widely used yet. It's still in development. I guess how to actually make this work is under development, but it is very promising, and it's really going to be great when we get it working, because it's pretty common to have those types of xenobiotics end up contaminating the groundwater, and that is anoxic. So you can't use Burkholderia to bioremediate any type of groundwater. There's no oxygen for it. It has to be something anaerobic that you would use. So that's for the xenobiotics. For the plastics, the situation is much more dire, I guess, or just a lot less promising. We really haven't made any progress on finding microbes that are able to bioremediate plastics and degrade them. Some of those plastics can essentially last forever in the environment. Of course, they'll be broken down into smaller pieces by kind of physical processes, but that's not necessarily great either, because then you end up with these itty-bitty little pieces of plastic that can be eaten by different animals and end up having negative health effects for them. So probably what will be successful to help our plastic pollution problem would be developing microbial plastics. A microbial plastic is a plastic that is made not out of a xenobiotic, but out of a natural compound that is produced by a microbe. So we actually have some plastics like this already. The most common one and most successful one would be the PHA-based plastics. PHA stands for polyhydroxyalkanoate. It's a polymer, of course, since it's going to be used for a plastic. And you have some bacteria that will actually manufacture the PHA polymer and use it as a way to store nutrients. So PHAs can be, depending on how you process them, they can take on kind of a lot of different physical characteristics in terms of like their flexibility and their strength and stuff, their moldability, which makes them useful as plastics. They have a lot of the properties that plastics have. And actually in Europe you have these things on the market, so it's possible to buy products that are packaged using PHA-based plastics. Then when you throw that stuff away, if it ends up in the environment, these same bacteria that made the PHAs in the first place can degrade them as well and use them to get energy. So the plastics will not just be sitting there till the end of time. This hasn't really penetrated the plastics market yet, of course, because of cost. So far, it's more expensive to produce the PHA-based plastics than to produce the regular xenobiotic-based plastics. And of course, with our economic system, if it's more expensive, it doesn't really matter. If it's better for people, it's just not going to happen. So, inevitably, as time goes on, these processes do become cheaper, and hopefully one day it will be cheap enough to replace xenobiotic plastics. This diagram here is showing a sequence of reactions that Burkholderia would perform to dechlorinate and bioremediate an example of a chlorinated xenobiotic. So here's your chlorinated xenobiotic. It has 3 chlorine atoms that have been added to it. Burkholderia will catalyze a series of reactions that remove 2 of those chlorine atoms from it, which makes it less toxic already. But then it's also able to break it open and metabolize it as an electron donor, which releases the last chlorine atom and produces succinate and acetate, which are not toxic at all. Next, we'll go over water treatment and water purification. So first we'll go over the treatment of wastewater, which is really important because usually we all get our water from the same source. If we're living in a particular city or town, then we're all getting our water from the same source ultimately. So if something is not right with that water, then we're all going to get sick, or we're all going to get poisoned at the same time. So in the past, when we didn't really understand necessarily so much about how disease is transmitted, or once we did understand that, but we didn't understand how to keep our water sources clean, it was actually really common for a lot of people to end up poisoned or sick at once because they all drank from a bad water source. Anytime that you're releasing wastewater into the environment, even if you're not, just turning around and directly reusing it as your water source, it will still come back to bite you. Depending on how the wastewater release is kind of arranged vis-a-vis your water source, you might have a lot of that wastewater ending up in your water source, or you might have a little and then it's just mostly being released into the environment. But all the water on Earth is actually connected, so things that you release in one place will end up affecting things in other places. In addition to the potential negative health effects on us, if we have poor wastewater treatment, of course you can also get environmental degradation if you're releasing untreated wastewater into the environment. So it can have really negative effects on the quality of the lakes and streams and ocean. It can kill fish and aquatic plants, and those things do end up affecting us as well indirectly, since we like to eat fish and stuff like that. You've got two types of wastewater. You have domestic wastewater and industrial wastewater. Domestic wastewater is also called gray water, and that's what comes out of your home, including your sewage. Also comes from restaurants or other small businesses. So most of the wastewater that's produced by average citizens would be domestic wastewater. It has a lot of organic compounds, especially because it includes sewage, and it can also have toxins that are coming from household cleaning products or stuff like that. But it's usually not too toxic and usually not too dirty overall. Well, at least not compared to the industrial wastewater. Industrial wastewater is coming from a factory or some form of industry, and it tends to be really dirty. It can have a lot of different stuff in it, a lot of different toxic compounds. What it has exactly depends, of course, on what type of factory it came out of or what type of industry it came out of. So you might have water that's really high in a particular type of toxin or in a different type of toxin from a different factory, but pretty low on organic compounds. Or if you're talking about water that's coming from an agricultural operation or a livestock operation, that type of water tends to be really heavily polluted with organic compounds and might not have as much other toxins in it. If the industrial wastewater you're producing has a really high amount of some type of toxin in it, it's really, really toxic, then the EPA mandates that you must pre-treat that before you release it to the normal wastewater treatment process. Basically because that process is not designed to handle the extreme levels of toxin that are present in your wastewater, and they would just pass on through being not treated or not completely treated. So the EPA's role there in enforcing that is actually pretty important. Otherwise, we would have a lot of really toxic stuff making it through the wastewater treatment process and, probably poisoning us. One way of kind of measuring the pollution level in wastewater is by using the biological oxygen demand, or the BOD. The biological oxygen demand is how much oxygen is required to oxidize all of the electron donors that are present in a water sample using aerobic respiration. So that includes, of course, your organic compounds that would be used as electron donors, but it also includes certain inorganic compounds that could be used as electron donors by aerobic chemolithotrophs. For gray water, usually it would have around 200 BOD when it starts, before it's been treated. For industrial wastewater, it varies like a lot. It might be really low, but it might be really high. Especially if this is an agricultural or livestock operation, then it can be up to around 1500 BOD. So very, very polluted with electron donors that would be used by microbes for their metabolism. When you're treating wastewater, the goal is to get rid of toxic compounds or to react them to some non-toxic form, or at least a less toxic form, and then to remove electron donors, both the organic ones and the inorganic ones, but of course most of them would be organic ones. If you don't get rid of those electron donors, then when you release that water, you're going to have a microbial population boom that will destroy the water quality and might produce toxic byproducts as well. In general, as far as removing the electron donors, your goal there is to get the BOD of the treated water down to 5 units or even less. At the end of the treatment process, what you're left with is called effluent water. Effluent water is wastewater that has been treated, and it's okay to release it now into the environment, or sometimes it might be purified so that it can be reused or even drinking. Actually, effluent water or any effluent liquid just technically means the liquid that's coming out of some type of a process. So you're going to have usually multiple stages in wastewater treatment, and at each stage, what is coming out of that would be called effluent water a lot, even though the final, the final effluent water is, what would be actually released to the environment. So I guess the term is just a little bit fuzzy like that. I've just got a couple pictures here of examples of industrial wastewater. It's pretty significantly polluted with toxic compounds, and as you can see by the color, those are different compounds. So you would need a different process to remove those in all likelihood. So the first step in treating wastewater is called primary wastewater treatment. In this step, you're basically just getting all the big stuff out of the water. So you're going to release the water into a tank and just let it sit there. All of the items in it, any objects that are in that water or large particles will sink to the bottom, and then you allow the water to run off. Run off the top, that is. So that water that's coming off the top isn't going to have particulates in it. Actually, it might still have particulates in it, but they're going to be like really small. All the big stuff will be gone. And of course, any toxins or organic compounds that were dissolved in it, those are all still there. And this water that's coming off the top, it would usually actually be called effluent water. It is not done, it's not done being treated, it's not ready to be released, but it is coming out of one step of the treatment process, so it is the effluent for that process. Most wastewater treatment plants, have additional steps, not just the primary wastewater treatment, because it really is not sufficient to remove all the toxins and stuff that are in the water. But you actually do have a few crappy water treatment plants that just stop here. They just, get out the big solids and then just let it go. So that's not appropriate. That's not appropriate. Very few wastewater treatment plants are like that. Certainly not ours. Here you have a picture of an example of a primary wastewater treatment tank. So you have your wastewater in here, it's settling, stuff is dropping to the bottom of it, and then it's going to end up passing through this series of grates, and only the water that's on the top will be able to move on to the next step, which helps make sure that any solids that are in it are being removed, since they would have sunk and they wouldn't be on the top. After primary treatment, you have some type of secondary wastewater treatment process. It's really common to have a secondary aerobic treatment process, but depending on what type of water we're talking about and what is contaminated with, different processes might be used instead. The secondary aerobic wastewater treatment is what's used for domestic water, most commonly. In this process, you are finally introducing microbes. They're going to be aerobes, since this is an aerobic process, and the basic goal is to reduce the amount of organic compounds that are in the water by using these microbes to degrade them. So this doesn't work if the water that you're trying to treat has a really high BOD, like, for instance, the water that comes off of an agricultural operation. But this is fine for our domestic water that has a more moderate BOD. There's multiple methods that count as secondary aerobic treatment processes. The most common one is the activated sludge treatment process. In the activated sludge process, you have the water that's coming out of the primary treatment process introduced to a tank, and the tank is always being mixed. So while the water in the tank is being mixed, which means that oxygen is being incorporated into it, much like if you're beating cream to turn it into whipped cream. You're incorporating air into it by mixing it. So it's the same thing here. You're also going to introduce specific microbes into this water that you want to be there, in particular zoogloia microbes. The zoogloia, when they grow, they form these things called flocks, which is like, basically it's like a bunch of slime. Yeah, so they form these slimy flocks, and then any compounds that are in there, like organic compounds, they're going to end up incorporated into the slime. Other microbes that are in the tank are also going to get stuck on the flocks and incorporated into them. So you're just kind of growing flocks in this mixing tank, and also making sure there's plenty of oxygen around for all these microbes to use for their metabolism. So the flocks are actually the activated sludge here. We're calling it sludge because it's nasty, and we're calling it activated because you have microbes in it that are actively metabolizing. So the zoogloia in there and all the other microbes that have gotten caught up in these flocks are going to be using the oxygen that's been mixed into that water to metabolize the organic compounds, using them as electron donors and fueling their growth. After the mixing tank, you would pump all that water with the activated sludge into a settling tank, and let it sit there. The sludge is, heavier than the water, so it's going to sink to the bottom of the tank. Meanwhile, it's still got plenty of oxygen in the water, so you're still having metabolism going on in that sludge. It is still activated sludge. So you're still metabolizing the organic compounds out of the water. And you also just have a lot of them that have gotten kind of trapped in these flocks. So that all sinks to the bottom of the settling tank, and then on the top what you have is clear water that has a lot less organic compounds in it than it used to, and it's ready to be removed, and exit the treatment process. Usually when you have that clean water coming out of the treatment process, you're also again going to call it effluent because it's just been through a treatment process. You would probably do something to it to make sure that any microbes that are left over in it are kind of removed. So usually that means maybe they're going to shine a UV light on it, or else they're going to add chlorine to it as a disinfectant. Then for the activated sludge that is settled to the bottom of this tank, some of that, a little bit of it, is going to be pumped back into the first tank where new water is being mixed and aerated, and is sort of like the little basis for the culture that you're growing in that tank. So in order to make sure that you always have zoogloia growing in that tank, whenever you remove it, you just take a little bit of the activated sludge and put it back. Then the rest of that sludge, you got to do something with it. So that, there's a variety of things that could be done with it. Sometimes it's sent to something called an anaerobic digester, where you have more microbes, but anaerobes this time, digesting that sludge, getting rid of all the remaining organic compounds in it by converting them to carbon dioxide or methane, even though methane is an organic compound. So that's one thing that can happen. That's a pretty common thing to do with a sludge. In some places they just burn it, which is not great. Burning is not a decent way to get rid of a waste product. And in other places, they actually use it as fertilizer. So in the pictures here, up top, you have a flock. So these lighter areas would be the slime of the flock, and all the little black, like rod things that's suspended in it, those are the microbes. So a lot of those would be zoogloia, and some of them are other microbes that just got stuck onto the flock. Here's a mixing tank, or it's also called an aeration tank, where your wastewater that has come from primary treatment is being constantly mixed so that air is incorporated into it, and of course the flocks are forming. And then here's a diagram of the whole process. So here's your wastewater coming in from the primary treatment process. It goes into the mixing tank. It's constantly being mixed. You also have flocks growing in here. Then it's sent to the settling tank. Your flocks or your activated sludge will settle to the bottom of the tank, taking organic compounds with them. The water at the top would be clear then, and it will leave the treatment process. So this is your effluent. Then this sludge here, most of it will leave, to somewhere else, maybe to an anaerobic digester or to be used as fertilizer, hopefully not to be burned. And a little bit of it a little bit of it will be sent back to the mixing tank to start the cycle again and provide more zooglobia microbes for flock formation. So this process is pretty efficient. You can actually remove about 95% of the organic compounds from that wastewater so that you end up reducing the BOD by around 95%, assuming that you didn't start with water that was too high in BOD in the first place. So, you know, as long as you were using gray water, it should be fine. After the secondary aerobic treatment process, you can go into a tertiary anaerobic wastewater treatment process, which would be like the anaerobic sludge digester. That type of process, the anaerobic sludge digester, could also be used as a secondary process if you're doing industrial wastewater treatment. So if you're using this treatment process of anaerobic sludge digestion after you've done the secondary aerobic wastewater treatment process where you formed activated sludge, in that case, that's a tertiary process because it's the third one that you're doing. If you're using this for wastewater that's come from an industrial source that has a really high BOD in it, so a lot of organic compounds in it, then that's going to be a secondary process. So you would have done the primary treatment process on that water to remove any large solids, and then you're doing this secondary anaerobic treatment process. In any case, if you're doing this anaerobic sludge digester process, then you're going to be using something called a sludge digester or a bioreactor or an anaerobic sludge digester or an anaerobic bioreactor to finish digesting all of the organic compounds in your wastewater. So this reactor is basically a tank that has no air in it, or no oxygen at all, and it has a lot of different anaerobic microbes in it. You want a lot of different ones that have a lot of different metabolisms. So some of them will be doing respiration, anaerobic respiration. Some of them will be doing fermentation. Some of them will be doing 1-carbon metabolism. The goal here is basically that any type of organic compound that you add to this bioreactor is going to end up being digested by one or more microbes that are in there, ultimately to carbon dioxide and methane. So any organic that you put is going to be oxidized to and ending up as just methane and carbon dioxide. You'll probably have a number of different microbes kind of catalyzing different stages in that digestion from, complete complex organic compound to simple methane or inorganic carbon dioxide. Then what's really nice about the process is that the methane that you produce could actually be used to run the bioreactor itself. So you don't have to have like extra fuel for it or extra electricity. You can just use the methane that the bioreactor produces itself. So that makes it really cost effective and it's pretty awesome. You can also remove like a lot of organic compounds from wastewater in this way, which is why you can use it for wastewater that's coming from like agricultural operations that has an extremely high BOD in it. So it would be used as a tertiary process for domestic water or as a secondary process for really high BOD industrial wastewater. So this is an image of a sludge digester. It's closed, of course, because, no air can get in, so you actually can't see what it looks like inside of it. But here's a diagram of what it looks like inside of it. So it has all these different layers in here. The sludge, the activated sludge, if you're using it as a tertiary process, then you would be introducing activated sludge into it that had just come from the secondary aerobic process. So that's going to end up in the tank, and it'll settle to the bottom and form into two different kind of layers. At the bottom is really kind of dense stuff that's all done being digested. Just above that you have a bit less dense sludge where you still have metabolism going on and organic compounds are being oxidized to smaller and smaller bits, ultimately methane and carbon dioxide. Then you have the supernatant, which is just the water at the top. So this should be cleaned water now. So you're going to remove that as the effluent and That's probably going to be actually your final effluent water in a lot of cases. And then at the top you have this scum layer, which is actually not a good thing. You don't want that there. That's something that forms from anything basically that's in the tank that is lighter than water. So if you have a lot of oil in there, it's going to end up in the scum. And also, you hear about stuff like rubber bands and stuff ending up in the scum layer. You're basically trying to minimize the size of the scum layer, because if it gets too big, it's going to start interfering with the supernatant, and even could come out, with the supernatant, which would mean that that's not clean anymore. So different bioreactors will have different ways of getting rid of that scum. This one is just drawing it off the very top. Then at the top, you have gases. This would be the carbon dioxide and the methane that are ultimately being produced by all the anaerobes in the tank. So as they degrade organic compounds further down in the tank, the gases they release are going to rise to the top, and then you have, a way to collect them up at the top. And this is just a summary of the reactions that are occurring, where you start with large organic compounds, you break them down into their building blocks, and then you're going to have fermentation and 1C metabolism playing a role to finish breaking these building blocks down into simple methane and carbon dioxide. Finally, we'll talk about advanced wastewater treatment. Technically, any tertiary wastewater treatment process is already counting as an advanced wastewater treatment process. So if you had your domestic gray water that was treated in the primary process, and then in the secondary aerobic process with the activated sludge, and then it went to the tertiary anaerobic sludge digester, that tertiary anaerobic sludge digester is now counting as a form of advanced wastewater treatment, simply because it's a tertiary process. So advanced wastewater treatment could be defined as any wastewater treatment process that goes beyond secondary treatment and produces water or produces effluent that is cleaner than what comes out of the secondary process. There's a lot of different advanced wastewater treatment processes out there that would be used in different circumstances depending on what's polluting the water in the first place. So there's going to be different goals depending on the process, but they would include getting rid of any remaining organic compounds, like with the anaerobic sludge digester, also getting rid of inorganic nutrients, prominently nitrogen and phosphorus, and getting rid of various toxins. If you're trying to get rid of phosphorus out of wastewater that's pretty polluted with phosphorus, theoretically, you could use microbes called phosphorus accumulating organisms, or PAOs, which take up phosphorus from their environment and incorporate it into a large polymer called polyphosphate. This is not really actually widely used yet because we don't fully understand how to make it work. We just need to know more about these PAOs. So we actually know remarkably little about them. But as we learn more, we'll make it work much better. The main challenge right now is making sure that the PAOs don't have stiff competition from other microbes also living in the tank with them. Then for nitrogen, you can remove that in a couple of different ways using microbes. You could use nitrifying bacteria and denitrifying bacteria sequentially. The nitrifiers would first be converting ammonia to nitrate, and then the denitrifiers would be converting nitrate to molecular nitrogen and also some nitrous and nitric oxide. So usually if you have nitrogen polluting wastewater, it would be either in the form of ammonia or nitrate, some type of fixed nitrogen. Those can both be removed if you're using nitrifiers and denitrifiers. The issue is that, of course, you're producing greenhouse gases. The other way to do it would be using the Anammox bacteria, which remove ammonia and convert it to molecular nitrogen. So there's nothing toxic about that product, which is good. The issue there is that only works for converting ammonia to molecular nitrogen. It doesn't work for nitrates. Then for other compounds like different toxins, There are certain microbes that can remove various toxins from wastewater. It really depends on what the wastewater is polluted with. Um, so there's some that can degrade, products or chemicals that are in household cleaning products. There's some that can degrade certain pharmaceuticals, and others as well. A lot of these toxins are xenobiotics, which makes it kind of hard to find microbes that will remove them. But actually in El Paso, our own wastewater treatment plant, which is really state of the art, actually, it's really strong. It uses advanced treatment processes that include using microbes to degrade some pharmaceuticals, such as certain antibiotics and stuff, which is really good. We're not all supposed to be taking each other's pharmaceuticals, of course, but when we do, do that, when we do take pharmaceuticals, maybe some people end up flushing some pills down the toilet, or a lot of those pharmaceuticals will be excreted in urine or in feces. So those compounds are entering our wastewater, and we like to reclaim that water because we live in a desert and there's no water around. So it's really important to be able to remove those pharmaceuticals so that we're all not just taking each other's drugs. Actually, I'll also just note here that birth control pills, birth control, like that medication, is secreted or excreted in the urine, so it ends up in the wastewater. And if you're not using some advanced process to remove that using a microbe, it's going to end up in the effluent and being released to the environment. So that means, potentially recycled and drank in by other people, but definitely, animals in the water will be exposed to it. So it actually turns out that a lot of these birth control compounds are active not just in people but in animals, and they're actually causing feminization of fish and other aquatic animals, because they are basically analogs of female sex hormones, which is in general a problem. Then we'll talk about drinking water purification. So the effluent that comes out of the wastewater treatment process is not drinking water. It is not safe to drink. So it's not potable. Potable means that it's safe to drink. Once you're finished treating wastewater, that's safe to release it to the environment. It's not safe to just turn around and drink it yet, though. So in order to make that water safe to drink, or in order to make any water safe to drink, no matter what the water source is, you have to do water purification. The introduction of purification of drinking water has had a really, really huge impact on public health and disease. It's dramatically reduced the rate of waterborne disease. Before you had, drinking water purification, people would just be kind of drinking whatever, and they were not drinking potable water. It was not safe to drink, and they did get sick from it quite a lot. So that's no longer a thing that really happens so much, and that's thanks to our drinking water purification. You always have two basic stages in purifying drinking water: first filtering it, then disinfecting it. For filtration, you've got three kind of steps of filtration that you will do. First, you have sedimentation. So you're going to have the water come into a tank and just let it sit there. Any big particles that were in it, we're going to settle to the bottom of that tank, and then you draw the water off the top, and it goes to the next tank, which is the coagulation tank. In that tank, you're going to add some chemicals to the water that cause any little particles that are left in there to kind of coagulate, which means stick together. So they're going to form larger particles. You're also going to have microbes sticking to them, and when they get big, when these little clumps get big, then we call them flocks. So you're forming flocks in the coagulation tank, and those flocks are going to sink to the bottom of the tank and stay there while you draw the water off the top. Then that water is going to go to the next tank, which is the filtration tank. We are actually going to pass it through a series of filters that are going to remove the very tiniest particles from it, and they also take out a lot of microbes and even some solutes too. So any particles that were small enough to get through the other stages, the sedimentation and coagulation, those are going to be filtered out, and a bunch of microbes as well. And then depending on which filters you're using, you might get solids also. if you're using like an activated carbon filter, then you're going to have some solutes sticking to that activated carbon and being removed from the water. And it is pretty common to use an activated carbon filter. The smallest microbes are going to get through that filter. You can't filter them out like that. And that includes viruses. Like most viruses are too small to really be filtered out, so they would get through that step. So because you have microbes coming through the coming through the filters and still being in the drinking water, you have to do another step after filtration, which is disinfection, just to get rid of any remaining microbes that are there. You can use a lot of different things to do the disinfection. You could use UV light. Technically, you could heat the water as well under pressure to disinfect it. You could add various chemicals to it, but it has to be something that's safe for people to drink. So even though there's a lot of options, usually we use chlorination, just adding chlorine to the water. UV is also used pretty commonly, but mostly just chlorination. And you're always going to do a couple of different disinfection steps, no matter what you're using to disinfect. Primary disinfection, then secondary disinfection. Both of them, of course, usually just by chlorination. So in primary disinfection, you are chlorinating the water, adding chlorine to it, and that is toxic to microbes, so microbes will start dying. Most of the microbes that were in that water will die. You're not going to get literally every single microbe dead by chlorination, but most of them will die. Also, if there's any organic compounds that are still left in this water, those are going to be oxidized by the chlorine. And that process kind of consumes the chlorine, so that the amount of chlorine that you added when you initially did the primary disinfection, that's not the amount of chlorine that's left in the water when the primary disinfection is over. You're not going to have very much chlorine left over after this process. And that's the reason why you do the secondary disinfection, in which you just add more chlorine. You basically are just adding enough so that when the water leaves the purification plant, it still is chlorinated. That way, if it's going through the pipes and it picks up more microbes, the chlorine is going to kill them. You want to make sure there's enough chlorine left in that water to kill any microbes that are introduced to it in the future. And that's the basics of water purification. You are going to have some microbes that do get past the disinfection step just because they're resistant to chlorine. Usually those are not pathogens, so it's usually okay. Here's kind of a diagram summarizing the purification process. So here's the sedimentation step where you add water, to your sedimentation tank. Large particles settle out. You draw water off the top. It goes to the coagulation tank, where you're going to be introducing some chemicals that cause flocks to form. They sink to the bottom. You draw water off the top. Then it goes to the filtration tank, where you're actually passing water through filters that remove any remaining particles, plus a bunch of microbes and solutes. Then you have disinfection. So you're going to have a chlorination tank where you are doing primary disinfection first to kill most everything that's in there, and then you would follow that up with secondary disinfection. Then you release that water, and that's considered potable. So you might store it and then distribute it. And then here's an image of an actual water purification plant with the sedimentation tanks, the coagulation tanks. The filtration is in this building right here. So when water is going through or coming out of this coagulation tank, it's going to pass through filters in these buildings and then go into these tanks here where it's going to be chlorinated. So this is all water that's coming right out of this river. So whatever wastewater is released into this river, treated or not, that's coming into our water purification plant here. So hopefully that wastewater was treated. The final kind of part of the water system is the water distribution. So after you have purified your drinking water and it's now potable, then you have to, distribute that to homes and businesses. So you're going to have a series of pipes to pump it and storage tanks to store it. Ideally, you would like to have no microbes in those pipes and tanks, but in reality, you do have some microbes. So you would have eliminated most of the nutrients from this water that would be available to support microbial growth, but just the truth is you can't do anything 100%. So there's going to be some nutrients left over that can support a small amount of microbial growth. Also, you have microbes that are resistant to chlorine. Those aren't going to be killed. Probably they'll still be in there. And as you're going, further and further from the initial purification of the water, both in terms of distance and in terms of time, the amount of chlorine that is left in that water is going to be going down and down and down. Then finally, you're going to have biofilms forming on the walls of pipes, especially. Well, pipes and tanks, but especially pipes for some reason. Most of the microbes that are in this water distribution system would be living in those biofilms on the walls of the pipes. And if they're in the biofilm, then they're going to be protected from chlorine. The chlorine cannot penetrate the biofilm. So as long as they're in there, they're safe. And then you're going to have inevitably little bits of kind of the surface of the biofilm breaking off, little microbes being released from the surface of the biofilm into the water. Some of those will be killed by chlorine, some of them will not, some of them will end up, in your drinking water. Most of the time it's okay. Most of those microbes that are in there are not pathogens, they're not bad for you, it's okay. But a few of them could be sometimes. So you don't really have any serious pathogens in there that are crazy hardcore, but you do have some that are opportunists or opportunistic pathogens. An opportunistic pathogen is some type of pathogen that would not cause disease in a healthy person, but could cause disease in someone who's been weakened for some reason. Maybe because they're very young, or they're very old, or they're immunocompromised, or they have some other type of serious chronic disease. So the fungi that we talked about before that cause mycoses, that mostly affect immunocompromised people, those would be classed as opportunistic pathogens. So yeah, there is some potential for people who are at risk to be damaged, or, to be sickened by microbes that are living in the pipes of the drinking water. To be honest, it's hard to know exactly how frequently that happens. Usually when someone gets sick, we never really find out exactly, where they got sick from. So it's known that this is happening. How much it is happening, that is not known. In addition to that, you have a good amount of protists living in the water, in this potable water. They're not going to be pathogenic to you. They're mostly there just to eat the microbes, the other microbes that are there. So they're predators, eating the different bacteria living in the pipes. But some of these protists will actually have bacteria living inside of them as parasites, which could include opportunistic pathogens as well. So that's another kind of way for opportunistic pathogens to survive in the water distribution system, inside of a protist. For these opportunistic pathogens, the genera that are most commonly identified would be Mycobacterium, Pseudomonas, and Legionella for the bacteria. For the protists, you have also a couple of opportunistic pathogens for people that are found in water distribution systems, which would be in the genera Naegleria and Acanthamoeba. And this image is showing a couple of protist cells that are infected with opportunistic pathogens, specifically Legionella. So all these kind of dark little blobs inside the cells, those are the Legionella bacteria that are living inside of this protist. And we'll conclude by talking briefly about the microbiology of buildings. So all buildings have microbes in them, and they're essentially living everywhere in the building. So on all the surfaces, in the vents, in the pipes, in the air, on the dust, everywhere. Most of them will be living on a surface, not just floating in the air, but there are some floating in the air as well. Most of them are going to be fungi and bacteria. You don't have very many archaea or protists living in buildings or algae, but that's not to say that you couldn't have any, just it's mostly fungi and bacteria. For the bacteria that you find indoors, they have usually been shed either by a person or by a pet, which means that they are related to the microbiome of that person or that pet. Actually, if you analyze the composition of the bacterial community within a person's home, you can identify or you can predict what pets they have, even if you don't know. And you can also see how people are kind of related to each other. So people who are in the same family will usually have similarities in their microbiome, and that means that they will have similarities in the bacteria that live in their homes as well. Most of the bacteria would be living on surfaces that you touch a lot. So, switches, doorknobs, handles, those types of things. Those are the surfaces that accumulate the highest amount of bacteria, as well as, of course, the kitchen sponge. And in general, if you have more ventilation in a building, more kind of air communication with the outside, then you're going to see more bacteria that have come from outside rather than coming from, a person or a pet. For the fungi that are inside buildings, usually they're not going to be coming from people or pets. Usually they come from outside instead. So the composition of the fungal community inside a home would pretty much resemble the composition of a fungal community outside that home. The only exception is there's some molds that are more common inside than outside, which would include like bread molds like rhizopus. So, fungi that are eating the food that you have in the home. Mostly these are not so dangerous for us, since they've come from ourselves or from our pet's microbiome. Usually we're not talking about pathogens, and most fungi are not pathogens, so usually that's pretty safe. Of course, it is possible to bring in something that would be pathogenic and, less safe. Why, we've been worried about that with coronavirus for months. We're practically living in BSL-1 conditions. But usually it's not dangerous. And then a final point to make about microbes in buildings is that anytime that you have microbes living on metal or on concrete, they're going to be producing waste products that increase the corrosion of those materials. So in general, it's good to take steps to limit the growth of microbes on buildings because they damage the structure, and which just costs money to fix it. Some of the ways that they could do that would be by producing waste products that are either oxidizers or reducers, or corrosive in some other way, or by altering the pH. Like there's some microbes that create an acidic environment through their metabolism.